Thank you all for coming. It's a great honor and a pleasure to be able to present some of my work here today. And what I've chosen today is to speak about something completely different and novel. And in general, our laboratory is involved in understanding the molecular mechanisms that contribute to oxidative stress and disease processes that result in things like age-related hypertension, atherosclerosis, radiation-induced vascular damage. But what I'm going to show you is how do we advance? Nope. Wait. Did something. Advancing the. There we go. Oops. Takes a bit of time. Is something completely different, and it relates very much to what Jen has actually presented to you, finding new things, different receptors in strange places. And this was, for us, a completely serendipitous finding, and I like this definition of serendipity, finding something cool without looking for it, or really a pleasant surprise. And for us, this has really been a pleasant surprise, which we've been able to follow. By way of introduction, what I'm going to talk to you a little bit about is blood vessel function. And interestingly, as David's spoken to you about the heart, the blood vessels are as important in the cardiovascular system, potentially more important because, in fact, the heart is a slave to the blood vessels. And as David said, many of the therapies that are directed in terms of the treatment of heart failure are actually directed at the blood vessels themselves. And so the blood vessels exquisitely regulate the flow of blood for the demand. So if we exercise, we know that there is the release of many mediators and hormones such as adrenaline, which mediate blood flow and dilatation, say, to the muscles. And there are mediators that are released by the very intricate and complex structure of the blood vessels. The blood vessels have a lining, cells called the endothelium, and the endothelium releases many mediators, one of them being nitric oxide, which act on the underlying vascular smooth muscle, which are also regulated by the sympathetic nerves, which release adrenaline. The sympathetic nerves, for the most part, cause constriction of the blood vessels. The endothelium, for the most part, releases mediators that lead to relaxation of the blood vessels. And so using classic physiologic assays, we actually can study the function of these blood vessels outside of the body. And the way that we do that is by actually isolating the blood vessels, cutting them into strips, and then we actually place them between stirrups and are able to we are able to measure the force that is generated by these blood vessels on a monitor by a force transducer. A force transducer converts, let's just go back. A force transducer converts the mechanical signal into an electrical signal. And here you can see as the blood vessel constricts, there is an increase in force, and as the blood vessel dilates, one sees a decrease in force. And I'm going to use this in many of the bioassays that we actually have shown. And so this is the serendipitous finding that my very astute postdoctoral fellow was able to observe. And the reason he was able to do that is we moved from a laboratory in which we were able to flip the lights on and off when we moved into a room to a room in which we had motion sensors which switched the lights for the laboratory on and off. And here you can see our myograph, which is our assay, and we've got a little blood vessel on there, and we induce, and we induce constriction of the blood vessel. We induce constriction of the blood vessel with small molecules like adrenaline in order for us to see vasodilatory responses. And as you can see, as he walked in the lab, he observed this small blip in the screen, which activation of light caused a relaxation of the blood vessel. And so we went back to the literature, and we're actually a little disappointed to find that this phenomenon of photorelaxation had actually been described by Furchtgott, who received the Nobel Prize for understanding the function of the endothelium approximately 50 years earlier. But we persisted and looked through the literature a little more and understood that, in fact, the mechanisms underlying this phenomenon had really been poorly explained, and that really no one understood why this had happened. And so we came up with a hypothesis, a kind of a little bit out of the way hypothesis, that this phenomenon may actually be mediated by light receptors, or opsins, we call them opsins, light receptors, which are localized in the blood vessels in a similar way to the way that olfactory receptors may be on endothelial cells, as Jen has explained to you. And so we pursued this hypothesis, and what I'm going to do is to show you some of the data that supports the hypothesis. I think we have a number of lines of evidence that this is indeed the case. Just by way of introduction, there are what we call visual opsins, and these are the receptors which respond to light, which are found in the retina. And those are the rods and the cones. And in fact, if we look, if we take a look at the rods, we find these receptors which respond to light. There is a chemical called cisretinol, which is a derivative of vitamin A. And when that sees the light, it actually changes its conformation, and that leads to a signal which actually allows us to see or to convert the light into an electrical signal, and we actually see a visual representation. However, there are also these non-visual or non-image forming receptors which are found in the retinal ganglion cells. And we know that blind people, people that don't have vision, are also able to or are able to have what we call a circadian rhythm. So they maintain circadian rhythmicity, which is important in our sleep-wake cycles. And so we hypothesized that it is these non-visual opsins which might be mediating this effect. And again, these opsins are incredibly ancient receptors. And you can see there are many, many different forms of the receptors. They're found in mollusks, because light is such an important signal with regard to motion and attraction. And so there is this tremendous, if this is the phylogeny, and it's even more complicated, this between these animals and going up to man. And so this is actually our bioassay. And as you can see, there is a light intensity dependent ability of the blood vessel to relax. And this is represented in the cumulative data over here, where we have the percentage of relaxation, how much relaxation there is on the x-axis, and on the y-axis. So the first thing that we wanted to do is to actually demonstrate, is this receptor present in the blood vessels? And we used classic molecular techniques to do this. We took the blood vessels, we isolated the messenger RNA, and were able to translate that to cDNA and use the polymerase chain reaction. And you can see that in the aorta and the brain, we have a signal of opsins, whereas in our mice that are deficient in this receptor, there is no signal at all. We then took mice, as I said, that are deficient in this receptor, opsin 4 knockout mice, and were able to show that when we exposed those animals, when the blood vessels of those animals were exposed to light, there was really no responsiveness to light at all as compared to the animals that had the receptor present. We were also fortunate in that a group had recently isolated, using a specific screen, small molecules that could actually block this receptor. And the reason that they were interested in this is that it is thought that the melanopsin receptor may actually mediate migraines. So we know that migraines are related to light intensity, and that in fact this may be the mechanism by which this is occurring. And so you can see in this data, again, we constrict the blood vessel over here. We see a light-dependent response over here. But when we add the compound, we see very, very little dose light responsiveness over here, and that's represented by the cumulative data here. Normal response, no response, or very little response at all. So the next question is, we know that these receptors are very have very specific absorption spectra. So they are very sensitive to different wavelengths of light. And we know that the melanopsin receptor absorbs maximally in the blue range of light. And so we asked the question using specific light-emitting diodes, which is a poor man's way of doing this experiment, whether there was a wavelength-specific responsiveness. And as you can see over here in the same bioassays, with a red light exposure, no vasorelaxation, green light exposure, no vasorelaxation, but in the blue spectrum, you see a significant responsiveness. And this is just, again, the cumulative data, and I actually have this is an ongoing trace in which we've constricted the blood vessels. It's going to take a little bit of time, but we actually see the responsiveness in real time. So I'm just going to let that run for 1 second, if that's okay. It can take us a second, sorry. I think it's worth seeing, though. So this is exposure of the blood vessel to red light, and you can see very little response of the blood vessel. There's green light over there, and then exposure to blue light should be coming up right now, since you can see the blue light. And if you actually see this quite profound vasorelaxant response. Now, we wanted to get a more accurate assessment of what the specific wavelength was at which the vasorelaxant response was occurring. And so we used what is called a monochromator, which actually creates almost a rainbow of different colors, of different spectra, and we can change the light wavelength in very small increments. And so what you can see over here is that the vasorelaxant response was maximal in about the 430 to 460 nanometers. And this is struggling with this. 460 nanometers, and you can see that this is exactly the response of the wavelength maximum at which the melanopsin receptor absorbs maximally. So this is, again, further evidence that the receptor is mediating vasorelaxant responses to light. We know that the blood vessel, the opsins that we are studying are what we call Gprotein coupled receptors. And Gprotein coupled receptors are classic receptors which transmit signals from the outside of the cell through a switch, the Gprotein, to an effector molecule, in this case adenylate cyclase in the cardiac myocyte. We also know that these receptors are regulated by brakes, which lead to a negative feedback system that Dave so elegantly spoke about. And so we asked, and again, this work was in 2012, Dr. Robert Lefkowitz and Brian Kobilka received the Nobel Prize for working out the signal transduction pathways and regulations of these Gprotein coupled receptors, which are so critically important as drug targets. So again, we asked the question whether the normal regulatory mechanisms, which are regulated by these kinases called GRKs, are important in regulating the opsins in the blood vessels. And here, this is just to show you what a negative feedback system is. We have a thermostat, a heat generator, and then the heat, and that feeds back. In the same way, we have light acting on the eye and a feedback system that prevents an amplification of the system. And we all know that when you walk from a dark room into the bright light, that response is actually damped over a period of time. And that occurs at a very specific molecular level, in that what happens is when we normally do the signal, with normal signaling, light activates these receptors, and we have an effector through this Gprotein. In terms of regulation, this kinase leads to a phosphorylation of the receptor, a binding of another protein, which then prevents the binding of the Gprotein and uncouples the effector from the receptor. And so we asked the question whether this is in fact the case with our receptor in the blood vessel. And what you can see over here is that with each repeated light stimulation of the blood vessel, the response becomes less dramatic. In fact, after a number of repetitious responses, one gets no response at all. In fact, when you add an inhibitor, though, of the GRK2 enzyme, you see a massive amplification of the response, and indeed, you see no attenuation of the signal over time, suggesting that this is an important mechanism of regulation. Now, the blood vessel assays that we had used are what we call blood vessels in the aorta, and those are buried deep dark in the chest and really don't have an exposure to light, and therefore their relevance is questionable. And so we moved to a more important regulatory blood vessel in the mouse, and that is the tail artery. And you can see that the responses to the tail artery are exactly identical to what we see in the aorta. More interestingly, we were able to show that using laser Doppler, which is a method for measuring blood flow in the tail, you can see that in wild-type mice, when we switch on blue light, you see about a 25% increase in tail blood flow and a reduction in that tail flow when we switch off the blue light, whereas in OPSIN4 knockout mice, we see absolutely no responsiveness to light whatsoever. So what are the relevance of all of this? Well, we know that vascular disease is prevalent. It's a profound cause of morbidity and mortality. But where can we potentially have impact where light might actually alter vascular reactivity in disease processes? And the one that we've initially started to think about studying is Raynaud's phenomenon, which is a profound enhanced vasoreactive response to temperature, particularly cold, in which there is a painful constriction of the blood vessels, an amplified constriction of the blood vessels. And so we're very interested in potentially using or leveraging this idea of light to treat Raynaud's phenomenon. And this, you can see, is just using laser Doppler to measure blood flow, and the red is a lot of blood flow, the blue is little blood flow, and this is a patient who has a moderate and then severe Raynaud's phenomenon. You can see very little blood flow at all. And so our idea is to add wavelength-specific light-emitting diodes to gloves to actually treat this disease process. And we know that pulmonary hypertension is a severe problem that leads to heart failure on the right-hand side of the heart. And there are limited numbers of ways in which we can currently treat pulmonary hypertension. So again, this is a potentially novel, safe way in which to treat vascular disease, which we hope to leverage in the future. And I'd like to just, again, also acknowledge the people that have worked on this, particularly my postdoctoral fellow, Dr. Sika. And I'm happy to answer any questions related to this. Thank you all.